Hi everybody, this is a very brief lecture on short-term synaptic plasticity. Short-term synaptic plasticity is one of the two major forms of synaptic plasticity, which describes how synaptic strength between two neurons or a neuron and any other target cell like a muscle cell can change in time, okay? And the short term basically means that whatever change that is happening doesn't last more than, let's say, a few seconds or a few tens of seconds at most. The long-term effects are usually more than an hour, and we're going to discuss those separately. The question of the plasticity of a synapse when it comes to short-term and long-term synaptic plasticity both is inquiring whether the strength of the synapse changes according to the history of the activity of the synapse. The simplest way to inquire this, which was a test that was developed by Bernard Katz and his colleagues when they were looking at the neuromuscular junction, is to inquire the output of a synapse with two presynaptic action potentials. So the first one that you do is the control, and you see a postsynaptic response. And then the second one is a test pulse, and you see if the second one produces the same response, in which the synapse is not showing plasticity, or if the response to the second action potential that is coming presynaptically produces a larger postsynaptic potential, in which case you have a phenomenon known as facilitation, or if it produces a smaller action potential, in which you have a phenomenon known as depression. These little blips that you see here are known as stimulus artifacts. When you stimulate the presynaptic cell, simply the electromagnetic field from that stimulus pulse produces a blip. So these are artifacts, they're little ones. The second thing that you see is the postsynaptic potential, and you see that the response here is facilitating. So facilitation is the increase in the strength of the synapse. Depression is the decrease in the strength of the synapse. And these are jargon that you need to know. Now, in this case, how do we measure facilitation? We can either take the ratio of the second response to the first response, which is called the paired pulse ratio, or you can ask how much is this increased with respect to that. So you take the amount of increase or decrease and normalize that by the amplitude of the initial response. And if you do that, what you can do is say, well, does this increase depend on the distance between these two pulses? If I increase that distance or decrease that distance, what do I see? So at the distance of 10 milliseconds between these two pulses, what you see is that if you plot that distance of 10 milliseconds and look at the amount of change, you see about a 30% increase over that, or about a 0.3 change over the initial response. That's what is plotted here as the amount of facilitation. If you make that distance shorter, go towards 5 milliseconds, then you see an even bigger response. You can't go less than 5 milliseconds because in this case, which is the squid giant axon, 5 milliseconds is pretty much the refractory period. You won't get to action potentials closer than that. But as you make the distance between the paired pulses longer and longer, then the amplitude of the response to the second pulse becomes closer and closer to that of the first pulse, and this amount of increase decays to zero. The reason for that is that facilitation or depression is due to something that the first pulse is doing. So the first pulse is producing a postsynaptic response, but it is also telling the synapse, I did something. Prime yourself for the second pulse coming in, and that is a form of short-term memory that this synapse is keeping. In this case, what facilitation is due to is typically accumulation of calcium in the presynaptic terminal that is not cleared fast enough. It often happens when the levels of calcium are low. So the first pulse, in addition to releasing some neurotransmitter, it increases the baseline calcium. The second pulse, it comes in, it produces more intrinsic calcium due to voltage-gated calcium channels, but because this is coming on top of some calcium baseline that was sitting there, there's a facilitation that's happening because the total amount of calcium is now bigger than the first pulse. But that calcium eventually clears if the distance between the two pulses is long enough, and this goes back to the initial level. The rate at which this goes back to its initial level is the rate of recovery from facilitation. Now, of course, when you have paired pulses, as I told you, and as you can see in this example in central synapses in the hippocampus, this is in the rat hippocampus in the CA3 area, if you put two presynaptic pulses, the synapse may facilitate in amplitude. Here, what you're seeing is synaptic current. Here, what you were seeing is synaptic potential. For this discussion, it doesn't matter which one we're looking at. In this other synapse that you see, the two presynaptic pulses produce a synaptic depression. The second pulse is actually producing a smaller response. Again, typically synaptic facilitation at the short term is due to accumulation of presynaptic calcium, whereas synaptic depression can have many causes. The primary cause is that you don't have too many primed vesicles ready to be released at that particular synapse. When the first synapse, when the first action potential releases the vesicles and the second one comes right after that, it doesn't have as many primed vesicles at the presynaptic terminal that it could bound, that it could fuse and release. Okay, so that means that recovery from depression requires more vesicles to be bound to the presynaptic terminal and release their neurotransmitter. Depression could also happen due to other factors. For example, you release too many vesicles and you saturate the postsynaptic receptors, and therefore the second time, even though you're releasing as much neurotransmitter, you may not have as much receptors that are ready to receive the transmitter because the neurotransmitter from the first action potential hasn't been cleared yet. But typically, facilitation is due to accumulation of presynaptic calcium, and depression is due to the fact that you don't have available vesicles to release when the second pulse comes. Again, if you have depression, as is shown here, you can plot the level of depression as a function of the paired pulse response. Here, only the response is shown. The action potentials presynaptically are not shown. So if these two pulses are coming close to each other, the first one is your control. The second one is producing a very small response on top of the first one, because again, you're not looking from the baseline, you're looking on top of this, how much is it producing. But if you have that 200 milliseconds later, it is producing a response that is about half the size, 500, it's still small. It takes about 5 seconds for the synapse to recover from depression. So if I take the paired pulse ratio now, the ratio of the amplitude of this one, which I measure from this point, to the amplitude of this one, which I measure from baseline to this point, and look at that plot, initially it's about only a quarter of the initial response, 0.25. But then it goes about 0.5 at this stage, and at about 5 seconds it goes back to the control value. Now, of course, you don't have to do your check on the presynaptic, on the synaptic plasticity this way with two pulses. You can check for the memory of this synapse a different way too. In this case, what we have is, just look at the top here. In normal calcium, we're looking at the amplitude of the synaptic response. 100%, it means normal amplitude. What we're doing is we're stimulating this synapse, let's say once every second, and if you do that, the synapse shows a fair amount of depression under normal calcium. So the amplitude, each dot is one inquiry, right? So the synapse is getting a little smaller, a little smaller, a little smaller, And then in this range, what we do is we produce a tetanus. A tetanus is a stimulus that comes at a high frequency, let's say 100 hertz. And you're stimulating the presynaptic cell at a very high rate, so once every 10 milliseconds, 100 hertz, for about 5 seconds. And what you see is that the synaptic amplitude rapidly depresses, right? It goes very, very low compared to control, which is 100%. And if you then go back and send your pulses, let's say once per second, it goes back. It doesn't go back to 100%, but it goes back to that slower depression that you were seeing. So you get a strong and fast depression due to a tetanus signal. All right, that's post-tetanic depression, right? Whereas if you reduce the level of calcium outside in the bath and you do the same thing, what you see is, in this case, you see the presynaptic response as a measure of the amplitude of the response, which is very small, but it's not changing when I send single pulses. When I put a tetanic pulse, in fact, increases the amplitude, and you get an initial facilitation, right? And then a slower depression on top of that, right? And after that, when I inquire, I have a lower level of response, but not much lower than what I had initially. So what I have here is indicative of the effect of the accumulation of calcium, whereas what I have here in normal calcium is indicative more of the fact that I'm running out of vesicles to release, okay? At much lower normal calcium, I only see the facilitation, which in this case they call augmentation. That's the same thing for facilitation for us right now. Augmentation is really facilitation happening at the time scale of seconds instead of time scales of tens of milliseconds. So this synapse is really showing two processes. One is depression, one is facilitation or augmentation. Now, whether you see depression or augmentation depends on how much calcium you have in the extracellular medium. And that means that these two processes, if I have normal levels of calcium, depression wins. If I have very low calcium, augmentation wins. If I have intermediate calcium, I have augmentation initially and then depression right after that. So you expose both of these processes. Augmentation or facilitation is due to accumulation of presynaptic calcium levels, whereas depression is due to running out of vesicles. The amount of depression that you get as a synapse depends on how much neurotransmitter release you have. So the relative amount of depression, if I plot that with the amount of neurotransmitter that I release, let's say if this is indicative of the number of vesicles, the more vesicles I release, the more depression I get, indicating that depression is due to running out of vesicles. Now, there's another form. So these are the forms of short-term synaptic plasticity that we get generally. We have depression, which happens at a time scale of tens of milliseconds to seconds. We have facilitation that happens on the time scale of tens of milliseconds. And if it goes in the time scale of seconds, we call that facilitation augmentation. And then we have an even slower time scale form of facilitation in which if you stimulate with a tetanic stimulus, so this is the postsynaptic response, we check it every second, let's say, and it's about 3 millivolts in amplitude in this case. Then we put a tetanus, we stimulate the heck out of it for about 20 seconds, and we stop. And after that, about a minute later, we see that the synaptic response is about 5 millivolts when we check it. And then it slowly recovers back to the original level over a few minutes. This type of facilitation that takes a long tetanus to expose and decays with a time scale of a few minutes is known as post-tetanic potentiation, or PTP. Post-tetanic, because the stimulus we produced was a tetanic stimulus or a tetanus, high-frequency stimulus. Potentiation is an even longer-term version of facilitation. The words facilitation, augmentation, and potentiation all mean an increase in the amplitude of the postsynaptic response. The word depression means a decrease. There's no three versions of it. So depression can be tens of milliseconds, can be seconds, or can be tens of minutes. We'll see that when we talk about long term. All of these things, which go on the order of tens of milliseconds to tens of seconds or minutes, are short-term synaptic plasticity. Now, in general, if you put a whole bunch of stimuli in a synapse, what you see is often that your postsynaptic response has a combination of responses. So initial response is this one, that's the initial amplitude of the response. And then the next one is larger, showing facilitation, next one bigger, bigger, bigger. And then you see the next ones are kind of smaller in amplitude. Again, we're measuring from the baseline here. These are becoming smaller and smaller and smaller, but we also have summation, which you learned about in the previous lecture. So this, what you see here, is temporal summation. Now, 30 seconds later, you have a postsynaptic response, which is much bigger than what you initially have, indicating the augmentation of this synapse. All right. Now, if I want to show the dynamics of this synapse in terms of what is happening, right, what I have is initially augmentation, facilitation, augmentation, which means it remains big, depression, and at this point, they call this potentiation, but let's forget that. The facilitation decays, but after 30 seconds, I also have, again, they call this post-tetanic potentiation if there's a tetanic stimulus, which I assume is this. Here I have augmentation or potentiation of the synapse. Okay, it's a complicated dynamics. I don't really care about you knowing everything. I want you to remember the words depression, facilitation, and potentiation. We're not going to talk about post-tetanic potentiation in this class, but we will talk about the words facilitation, depression, and potentiation themselves. Those words are going to be used in our discussion. As a behavioral example of the role of short-term synaptic plasticity, I'm going to give you this example from the sea slug, Aplysia californica. This is the giant sea slug that lives in California waters. And in fact, the study of this sea slug in understanding, learning, and memory resulted in the Nobel Prize in the year 2000 for Eric Kandel and his colleagues. So what the behavior is that I'm going to tell you about has to do with an organ named the siphon of the animal. So the siphon is connected to the mantle and the gill, and when the animal is in seawater, the siphon is out and it pulls out the gill with it and allows the animal to get seawater through the gill and breathe. However, this pulling out and pushing in the siphon and the gill is controlled by a set of neurons that are in a ganglion in the abdomen of the animal, known as the abdominal ganglion. It's part of the central nervous system. And the abdominal ganglion neurons can be identified easily because they have large cell bodies and you can record them. There's sensory neurons, there's motor neurons, and there's interneurons in the abdominal ganglion. Think of these as the equivalent of the spinal cord for this animal. Obviously, this is an invertebrate. Now, the behavior is the following. If you touch the siphon of the animal, it retracts it very rapidly. So this is the magnitude of the retraction when you touch the siphon. And then gradually after it retracts it, it lets it back out again, right? If you touch the siphon again and again, it keeps retracting it. But the amplitude of the retraction gets smaller and smaller. And then the animal eventually realizes that this touching is not a harmful stimulus, so it stops withdrawing the siphon. This change is known as habituation. That's a behavioral term. Now, if you touch the siphon and at the same time either pinch the tail with, let's say, a pair of forceps or shock it electrically, then you get a strong retraction of the siphon even after habituation. So that's, in terms of behavior, that is known as sensitization. So you have habituation, but if you pair it with a noxious stimulus, you get sensitization of that behavior. Sensitization is shown in the graph here. So here is the magnitude of the gill withdrawals. Gill withdrawal and siphon withdrawal are the same thing because they're connected to each other. So without a shock, the amplitude of the retraction goes down and down as you touch the tail. But if you give it a tail shock, then you get a much bigger amplitude, but then returns back to the baseline of the control. But if you give it multiple shocks, you can actually make the animal become sensitized to the withdrawal when you touch it, right? So you give it shocks and then you test it by touching the siphon. That's what you do, right? So you pair it and it withdraws, and if you touch it again without shock, it will also withdraw very strongly for about an hour. That's what is shown here. But if you give this pairing multiple times or multiple times over multiple days, then you can make the sensitization last for a long, long time. The long-lasting sensitization will come back in the discussion of long-term synaptic plasticity. Right now, I want to talk about sensitization itself. So again, you touch, it habituates, you pair it with a shock, it becomes sensitized so that even if you then touch it right then, without the shock, it will withdraw it immediately. Okay, so sensitization, testing that it's sensitized does not require the shock itself. Testing it just requires the touch, but sensitization itself requires pairing the shock and the touch. All right, so what is happening inside the nervous system? What I want you to focus on here is the sensory neuron now that is basically a touch sensory neuron connected to the siphon skin, and when you touch the animal, that sensory neuron detects the touch and it excites a motor neuron that withdraws the gill and the siphon. Okay, so this is a simple sensory motor reflex that you see here. Now, the tail itself also has a sensory neuron. This is a noxious sensory neuron or a nociceptor. So think of it as a pain receptor, right? So when you shock the tail, this sensory neuron becomes active. That sensory neuron activates a modulatory neuron that then acts to release a neuromodulator and change the synapse from the sensory neuron to the motor neuron. Ignore this interneuron in this discussion for now. So what you see at the level of the nervous system is that this sensory neuron, right, every time you touch the skin, the siphon, it produces an action potential. You go, it produces an action potential, but the EPSP that it produces on the motor neuron is initially large, but then it goes through short-term synaptic depression. As you touch the tail, it becomes depressed. When you stimulate the when you stimulate the tail, what you get is through the action of this sensory neuron, through this modulatory neuron, this synapse from this sensory neuron to the motor neuron becomes enhanced again. This is a modulatory induced facilitation of the synapse. And that increases the ability of this sensory neuron to activate the motor neuron. So it basically overcomes the synaptic depression. All right. How does this happen? So this is basically looking at the amplitude of the EPSP and what happens. I'm going to ignore this for now. But what is happening, I want you to focus at this particular synapse. And again, ignore the interneuron. There's a modulatory neuron coming in to the sensory motor synapse. And this modulatory neuron that is coming in releases serotonin. The sensory motor synapse itself is a glutamatergic synapse. So a simple excitatory synapse, it releases glutamate, excites the motor neuron, right? It's gone through depression. However, when the serotonergic modulatory neuron becomes active, it releases serotonin, and the serotonin receptors are on the presynaptic terminal. So this modulatory neuron is acting presynaptically through these receptors, and these receptors act to activate adenylyl cyclase. Adenylyl cyclase, as I told you, is a membrane-bound protein that converts ATP to cyclic AMP. When this happens, cyclic AMP is a signaling molecule that activates a kinase known as protein kinase A. Protein kinase A, when it's activated by cyclic AMP, the catalytic and regulatory subunits come apart. The catalytic subunits become phosphorylated. They activate both a set of potassium channels and a set of calcium channels. They activate a set of calcium channels and turn off, close a set of potassium channels. The effect of closing the potassium channels is that when action potentials arrive at the nerve terminal of the sensory neuron, they become wider. When they're wider, they allow more calcium entry through the calcium channel, and that produces more release of glutamate. So that's the simple sensitization action. In the longer term, that same mechanism, if you keep activating it and activating it and activating it, it actually starts that activation of protein kinase A. Because protein kinase A becomes activated by separating its catalytic and regulatory subunits, it becomes persistent. So pKa becomes persistently active, and that results in changes that go back to the nucleus and make gene expression through this protein known as CREB. I'm going to talk about this in the next lecture. And that becomes a more long-term sensitization. That part is long-term plasticity. The part that I wanted you to see right now is short-term plasticity, which you understood. The habituation is due to short-term synaptic depression of the sensory motor synapse, and the sensitization in the short term is due to short-term facilitation of the synapse simply by this modulatory action, by widening the action potentials through closing the potassium channels.